Hello, everyone. So, how's your day going after 2 days of the conference? I hope you have enjoyed it. I have personally enjoyed it, meeting with great people and seeing how great work has been done in this field. And my presentation is mostly related to the discovery that I've had. I'm a clinician and I practice oncology in California. The company that we actually have is called Research Cancer Institute of America. That's the company that we have designed around the protocol that I have invented. Pacific Medical Center of Hope is the clinic that we have, and we treat patients with very advanced cancer in that clinic. The therapy that I'm going to talk about today is called multi-targeted epigenetic therapy, or MTET. The reason that we called it multi-targeted epigenetic therapy is because the therapy actually works on epigenetic targets and beyond. We established the company around 2008. The company is growing very quickly since we established it. Back then, we have designed several experimental therapeutic concepts and as well as clinical data that we have collected. It was established in 2008. We have applied for the patents in the US as well as Europe and other countries, and we have been able to get approval from the patent office on several applications. We have a strong team, regulatory, financial, and scientific. As a clinician, we used to prescribe the medications based on what is approved. And unfortunately, clinicians are not usually involved with drug development. But what happened in my clinic was that as I was using the treatments mostly off-label and in combination, I figured that I have no other choice but becoming a scientist and drug developer, because the therapy was so effective beyond the standard treatments all across the solid tumors and hematological cancer that there was no other option for me. We started to work with National Cancer Institute as soon as we started the treatments, and we were able to publish many of these great results. NCI was reviewed the cases, and they had supported significantly the work that we've had. And after reviewing the cases, they approved the therapy to be novel and effective. We have, as I said, very strong preclinical work as well around the therapy that started with working with experimental therapeutics in different academia. And then further, we sponsored research in other institutions. The technology is around the small molecules that some have been repurposed and some have been combined into new formulations. Therefore, the patents are both for methodology as well as new composition of matter. We have data on phase 1 as we speak. We have phase 2 trial going. A wide range of safety has been proven for the therapy. It is active in a variety of the types of tumors, mostly epithelial cells driven, and also very effective in hematological malignancies. The therapy is synergistic with the standard therapies, all categories from the cytotoxic drugs to targeted therapies as well as immune therapies. So we have proved the concept in that as well, showing significant synergism between the standard treatments as well as our therapy in variety of types of tumors in different categories. So if you're talking about cytotoxic drugs, we have proved the concept on increased efficacy when combination is applied. You're talking about targeted therapies. Our therapy is able to reduce the resistance to the targeted therapies. When you're looking at combination with immune therapies, we've been able to prove that the therapy works much better. The immune therapy is working much better when the epigenetic treatment is applied. I should say that I'm not the only person who has claimed this. So if you look at the literature, you see that by the work of, for example, the the DREAM team, Stephen Bailey at Johns Hopkins and others, the concept of application of epigenetic treatments to the immune therapies and targeted therapies has been suggested. But what we added was to modify exactly what sort of epigenetic therapies are clinically relevant to be combined with existing drugs. Basket trials preferred for phase 2/3. So as you know, most of the trials that now are being conducted are based on the new understanding of the cellular biology. So instead of being tumor type specific, most of the trials are now basket trials. And so what we propose from this stage, instead of just type specific application on our trials, is to hopefully partner with the strategic partner that we have cultural alignment, and hopefully we can take this to this phase 2/3 trial, looking at the basket trials and looking at the specific biomarkers as we have developed. Companion diagnostics, we have worked on identifying what sort of companion diagnostics we would need for phase 2/3 trial that we are proposing. And so with the appropriate partner, we can share this under the CDA. Challenges of the current technology and products in oncology. So as I'm sure many of you might be aware, especially clinicians, I'm sure they're aware, that the current standard treatments, if it's a single drug, mainly fails, and therefore the combinational therapies have been proposed to maximize the efficacy. But the unfortunate reality about combination therapies is that when you combine two toxic drugs together, you may get increased efficacy, but for sure you're going to get increased toxicity. So that has been a big barrier, and especially as far as the regulatory barrier, many of the companies that are trying to do combinational therapies fail to bypass the toxicity question from the FDA. Our therapy shows that in combination with the standard treatments all across the board, not only the toxicity does not increase, but rather the toxicity decreases. So we have a large preclinical as well as clinical data to prove that the toxicity of the combination therapy is very manageable and even better than the standard single agent therapy. Majority of the targeted therapies activate feedback loops, and desired overall survival advantage is limited. I'll give an example for targeted therapies for erlotinib. As you know, erlotinib is being used for EGFR mutated lung cancer and also pancreatic cancer. The problem with using targeted therapies all across the board is that the feedback loops become active, and when that happens, the tumor becomes more resistant. The combination of our products or our technology with the current targeted therapies inhibit that. So when that happens, then you see that the tumor continues to respond to the appropriate targeted therapy that is selected. By reducing the resistance, the tumor recurrence is less likely. Also, immune therapies and cytotoxic therapies mainly activate the cancer stem cells. So when you're using the cytotoxic therapies, because of increased intratumoral hypoxia, the tumor stem cells become more active, and that's a reality all across the board. I'll give you an example. When you treat a patient with early stage or early diagnosis of breast cancer, the standard treatment is using adjuvant or neoadjuvant chemotherapies. But the reality is that every time you use chemotherapy, you put the patient at risk for increased stem cell activity and progression of disease. So clinically, I can give you examples that we see every day in our clinic, or I should say we used to, that we treat the patients with cytotoxic drug, standard care, and as the patient is receiving the treatment, the tumor spreads. So it's very hard to believe that the tumor that has been sitting in the breast for 5 years and has never spread, it just happened to spread by itself as soon as you started the chemotherapy. So that correlates and is consistent with the preclinical data confirming that cytotoxic therapies increase intratumoral hypoxia, they stimulate the angiogenesis and the progression of disease by metastases. Our therapy, in other hand, reduces the chance of tumor angiogenesis and the stem cell activity. So, multi-targeted epigenetic therapy, reduction in circulatory tumor cells and circulatory stem cells by inhibition of epidermal mesenchymal transition, improved surrogates of overall survival in a matter of 2 weeks. So this is a clinical claim. This is a clinical statement as a clinician I make, because 90% of my patients, when we start the treatment on them in a matter of just 2 weeks, which includes only 10 treatments on a daily basis, they have suppressed and 90% normalization of the angiogenesis markers, which are surrogates of survival. So as you know, circulatory tumor cells correlate with the patient's survival. When a patient comes with breast cancer, lung cancer, and colorectal cancer, by just doing a blood test in the US and in our clinic for all types of cancer, we send the blood to Germany and we look at the circulatory tumor cells. After 2 weeks of treatment, we were able to substantially reduce the circulatory tumor cells in the patients, and most of the time to 0. Mechanistics. So as I said, after I discovered this treatment about 10 years ago, we had to go to the lab and look at the mechanistics to find out what targets are there, what are the ideal basically biomarkers to look at for the patients. So every patient that comes to see me, we do a molecular profiling before we start the treatment. If we have the tumor available, we do it on the tumor. If we don't, we do it on a liquid biopsy basis. And we do that after the treatment. So we have a comparison before and after on every patient. We have shown that the mechanistics are independent, so the patient does not need to receive any sort of standard of care treatments. They can just use MTET therapy. And it's also synergistic, so you can use it with combination therapy. Obviously, you need to have the knowledge to know what sort of synergism exists between each drug to be combined. And when I talk about independent efficacy, it took us a long time. I would say about 5-6 years it took us to find out what sort of mechanistics are involved for independent efficacy. So when we're seeing these clinical results, we have to know what targets are affected, right? So it was very interesting, and it was a very long journey, because the target, as the name pertains, multi-targeted epigenetic therapy, it had so many targets. The mechanism of effect was through cytotoxic effect. It had targeted effect, it had T cell tumor specific response, and it had epigenetic effect. So just to explore what you're going to accomplish by using a multi-targeted therapy, you see you're using, by one treatment, you're actually using 4 categories, at least 4 categories of drugs at once. Now you tell me if you can, or any scientist who claims that he can combine 4 drugs in a patient, each from one category of conventional treatment. If you can combine 1 immune therapy with 1 targeted therapy, with 1 cytotoxic therapy, and 1 epigenetic drug, it's impossible, because even combination of 2 category drugs are toxic, let alone 4 category drugs, and yet have no toxicity, almost no toxicity of the combination. That is why all across the board we have such a success, because we're using 4 category of drugs at the same time with no toxicity. Key targets, as I said, the tumors that are more hypoxic respond better to our therapy. So hypoxia-inducible factor 1, vascular endothelial growth factor, phosphatidylethyl 3 kinase, BCL2, PARP, and PGP. Each of these targets are somewhat shared between different types of cancer. So during the Darwinian evolution of the cancer, what you call the heterogeneous nature of the tumor, you see that all these targets, somatic mutations, happen during the time and between the different metastatic lesions. So you have intratumoral heterogeneity and intertumoral heterogeneity that happens not only by selective pressure that you cause by using different treatments, but also by the nature course of disease. So the reality is that if you have something that goes after all these sort of somatic evolutions and targets the heterogeneity of the tumor, you got the holy grail, right? So it's superior to the current VEGF inhibitors. We have shown that our therapy is superior to that. We have shown that in combination with TKI inhibitors results in resensitization, as I explained. We have tested, as I said, all types of tumors. The ones that we have most data on are ovarian cancer, lung cancer, and glioblastoma, but we have significant data on, or I should say, all types of cancer. We talk about the most difficultly to treat pancreatic cancer, colorectal cancer. We have all that as well. So stem cell efficacy, we talked about that. Our therapy disconnects the microenvironmental support of the tumor. That's why it works on the snail slug targets. There is no available approved therapy for stem cell plasticity, as far as I know, except our therapy. It should be a backbone therapy for all other treatment modalities, as I said, because if you don't go after the stem cells, whatever you do besides that is going to fail over time. It has overall survival benefit. We're not talking about PFS. I'm running out of time, so I'm just going to go quickly over the slides. This is our therapy and effects on the hypoxia. Lung cancer, as you can see, the migration and invasion of the tumor. The first two are the control and the vehicle, and then after exposure to our therapy, you see there's significant reduction on the viability, migration, and invasion of the three-dimensional cell culture spheroids in the lung cancer. I don't think I have time to go over the cases, but I had prepared a couple of cases with CNS involvement, advanced stage 4 disease that were dying in less than 2 weeks, and I saved them as we speak. They are in remission. This is a PET scan of a patient that we treated with a stage 4 breast cancer with involvement of her brain, facial paralysis, and leptomeningeal disease. As you know, it's an incurable disease. Leptomeningeal disease means death sentence. There's no single case study has ever been published on treating patients with brain cancer with leptomeningeal disease with any sort of therapy. This patient, as we speak, has no signs of cancer. Tumor markers, as you can see, 1,000 when we started. Look at that. Scan, pre-post. This is before we started her under treatment. As you can see, the bone are lighting up like a Christmas tree in her PET scan. And then post-therapy is completely clean. Or I should say minimal residual disease. You really can't say if she has cancer. Dural disease, as you can see, this is the first MRI we got after 2 months of treatment. As you can see, the dural and leptomeningeal disease has decreased over 50%. Future mission, this is my last slide. Strategic partnership aimed at expansion of application, academic and industry. FDI and the applications for phase 2/3 trials, expanding target biomarker development. Improved delivery systems, including different cellular vectors. Protocol development for combinational therapies with gamma knife in GBM. That we have a trial that we are designing right now for that. IP advances in proof of principle for combinational therapies in both available drug and failed therapies in the industry. We have several drug candidates that they have failed phase 3, and we think that they would be great combinational therapy with our protocol. Exploring regulatory policies beyond the US with international collaborators. We have several interests internationally from other countries, including Middle East, that are interested to collaborate. And I think that's all. Thank you.